Hi everyone. In the past video I talked about the transcription, replication, translation of the genes and the different units, mRNA, tRNA, and all that stuff. In this video I'm going to talk about operons and more prokaryotic genes and what that looks like. So let's get into it. So again, last time we discussed how genes in eukaryotes, cells with nucleuses, are expressed, and the steps of transcription and translation. But in this video, I'm going to more transition into prokaryotes and the genes in these kinds of structures. So remember, prokaryotes are cells without nuclei, and so although they do not have a nuclei, they still have DNA. This DNA is just floating around in their cytoplasm, and it has a couple of things that makes it different from DNA in eukaryotes. So first of all, each of the genes in eukaryotes is controlled by one promoter. A promoter is a sequence before each gene that allows the gene to be transcripted, basically telling the polymerase it's okay to express this gene, and letting the polymerase create a complementary strand and so on. However, in prokaryotes, instead of each gene having its own promoter, there are multiple genes under the control of one promoter. This affects gene expression, as if one of the genes is not wanted to be expressed, then the whole set is not expressed. However, the genes are under a single promoter are related in terms of function, so this doesn't seem to be that much of an issue as they all function together. Further, in addition to the genes and promoter, there is another sequence above the promoter or upstream of the promoter called the operator. This basically acts as an on-off switch for the operon genes. Together, the genes, promoter, and operator are called the operon. So exactly how are these genes turned on or off? Well, it all depends on what actually attaches to the promoter. If the RNA polymerase attaches to the promoter, then the operator will turn the genes on, and they will be transcribed and later expressed. However, if a structure called the operon repressor binds to the promoter before the DNA polymerase can, it blocks the DNA polymerase from getting to the promoter, and thus the genes are not able to be transcribed. Another question: how does this repressor bind to the promoter? Is it some kind of race for the polymerase and repressor to get to the promoter? Not exactly. What really happens is dependent on the environment of the operon. In order for the repressor to bind to the operon, it needs a protein to first bind to it, and this is further dependent on if the cell needs the enzymes that the genes will code for. I know this all sounds kind of confusing, so let's take a situation. So suppose there is an operon that produces lactose. If the genes that are controlled by the promoter have functions dealing with producing lactose, but the cell has enough lactose, then the sequence above all this that codes for regulatory genes will create a protein, and this is the protein that will bind to the repressor and then will bind to the promoter, making sure that RNA polymerase does not transcribe the genes that will make more lactose. But suppose that the cell does not have enough lactose. Then the sequence that codes for the regulatory genes, called that because the protein basically creates or regulates expression, it will not create the protein, so the repressor cannot bind to the promoter, and the RNA polymerase will need to transcribe the lactose genes, giving the cell the needed lactose. So this is what I mean by everything is dependent upon the environment of the cell, and regulation will occur accordingly. Also, just to note that the structure of the repressor and the protein together is called a co-repressor. So the type of operon we just talked about is called a repressible operon, where the genes can be turned off based on the environment. But there is also such operons called inhibited operons. These are operons which have the default as off, so the genes will automatically not be transcribed, and need some protein to turn the operon back on. So these are like the opposite of repressible operons, which need the repressor to turn it off. The inhibited operons need proteins called inhibitors, which turn these operons on by inactivating the repressor that is sitting on top of the promoter. So that was it for this video. If you like this video and liked these kinds of genetics videos, then subscribe to this channel because I have a whole genetics video series, and there's also a bunch of other kinds of videos on this channel, such as computers and anatomy, book reviews, and all that sort of thing, so go check those out. If you have any questions about this video or anything else, then please leave those in the comments below. And if you have any ideas for future videos on academic topics, then also leave those in the comments below. Again, subscribe to this channel, give us a thumbs up if you liked it. And here's a couple of citations I used for the pictures for this video. Thanks for watching. Bye!